Hello and welcome to this afternoon's Science for All Seasons talk. The Science for All Seasons series gives you a chance to explore hot topics in genomics and biology with leading experts at the Broad Institute. My name is Namrata Sengupta. I'm the Associate Director of Scientific Public Engagement and Broad Discovery Center at Broad Institute. And it's my pleasure to welcome all of you here this afternoon and to introduce our speakers, Sonia Vallabh and Eric Minikel. Sonia and Eric are scientists who co-lead the initiative to develop preventive drugs for prion disease at the Broad Institute. They come to this work with a personal mission. Together, Sonia and Eric will share with you about their decade-long journey to understand the roots of prion disease and hopefully find a way to prevent it before it strikes. If you have questions for Sonia and Eric, please ask them using YouTube's chat feature, and we'll do our best to discuss them during the Q&A portion of the afternoon. If you're tweeting today, please use the hashtag BroadSFAS. Sonia, over to you.
Great. Thank you so much. And it's really a pleasure to be here today to talk with you all about our personal and scientific quest to hopefully develop a treatment for prion disease. So to cue up why we do this work, we have to go back in time a bit to our wedding. This is a photo of Eric and I in August 2009 getting married. And on the right is a photo of my mom helping me get ready that day. So she was 51 years old at the time. And I can't take any credit for organizing the party. That was all her. Energetic, super happy. It was a beautiful day with her. But what we couldn't have known at the time is that this was actually the last family event for which my mom was healthy and with us. It was only about 6 months after these pictures were taken that her health started to change in what seemed at first like minor ways, so changes in eyesight, unexplained weight loss. But week by week, things were snowballing and quickly getting out of control. So from minor symptoms in February, by March of 2010, she couldn't finish a sentence. She was too confused to really communicate with us. She was too weak to get around the house by herself. Her abilities were just falling away right before our eyes. It was scary to watch, and we were rushing her around to doctors, trying to understand what we were looking at. And nobody really had answers for us. All we knew was that in this sort of time-lapse video way, she was declining. And in June, she went into a series of hospital stays that actually she never came out of. In December, she passed away. And it was only after she died that we learned from her autopsy report that she had died of genetic prion disease. And embedded in this diagnosis was this whole new sort of vista for us, the terrifying knowledge that this journey wasn't over. And because this is an autosomal dominant disease, sort of like Huntington's disease, for example, I was at 50% risk of having inherited the mutation that had caused my mom's disease. So this put us at a crossroads, and we had to decide whether we wanted to get predictive genetic testing in order to learn what the answer was. Had I inherited the mutation or not? And we actually got a lot of advice around this time to not get predictive genetic testing. Prion disease is fatal and untreatable, and people with our best interests at heart advised us to consider walking away. But what we knew about ourselves, I think, was that we couldn't go back to a time before we knew that I was at risk. So we had already been sort of transported into this limbo. And the limbo itself was acutely stressful. We were waking up every day wondering. So it felt like there was no way except forward. Get the result and at least learn what we were up against one way or the other. And so that's what we did. And at the end of 2011, this is the genetic test report we received, and it wasn't the news we hoped for. So I did inherit the mutation from my mom. And the sort of highlights of what this meant then are actually still true today. So prion disease is fatal, it's untreatable, it's a neurodegenerative disease. my mutation and a handful of others in this gene actually convey very high risk of developing the disease in your lifetime, upwards of 90%. But the tempo of prion disease is a little unusual. It seems like people who carry these mutations remain healthy for a good chunk of years, decades, and then at some unpredictable point, symptoms begin. And from that point forward, what we saw with my mom, as sort of shocking and terrifying as it was, actually is not unusual. Most patients die within a year or even 6 months of their first symptom. So this was a lot to take in. And we actually weren't in sort of any special position to receive this information. We weren't trained as scientists at the time. I was actually trained as a lawyer, and Eric was trained as a transportation engineer. But this ended up being a pretty dramatic turning point in our lives. So what we realized sort of moving forward in the weeks after my test report was that we were highly motivated to learn more about this disease. It had come crashing onto the scene. It was going to be part of our lives, like it or not, going forward. So we felt like we wanted to know as much as we could, learn about it, figure out who was working on it. And having seen my mom's experience, we felt that this was a rare enough disease that we couldn't necessarily count on doctors to know if progress was being made and to hand us that information. We sort of felt like we had to be our own advocates, and so that meant becoming savvy consumers of scientific information ourselves. This started out pretty humble, sort of reading online, calling up researchers who would chat with us, talking to our friends who were scientists. But within a few months of receiving my test report, I had actually left my job and taken a new position, entry-level role in a research lab at Mass General Hospital. And Eric followed a few months after that and took a role as a bioinformatician. So these first jobs in science, I think at first were also sort of part of that quest to just educate ourselves. And I think when I first took that job, I didn't see it as this dramatic and permanent career change. It felt more like, this is the means I have to productively engage with this new information. And I need to do this right now, maybe as a sabbatical from my regular life. But after a little while in these new roles, we realized that we weren't going back, that we had what felt like productive momentum that was moving us forward in science. And this was our new path. After about 2 or 3 years in those first roles, we reached sort of another juncture where it was time to figure out how do we pivot from these jobs where we've been learning some of the tricks of the trade, learning what science looks like in practice, but not working on prion disease by day. How do we shift from that to actually working on our mission? We want to contribute to curing this disease. We need to figure out how. And this was sort of an interesting assessment. There were a lot of different possibilities. We had started a nonprofit, Prion Alliance. Should we go full-time on the nonprofit? Some people advised us to think about starting a biotech. And I think every disease has sort of this different constellation of knowns and unknowns and assets and gaps. And it was really a sort of disease-specific analysis that we did, looking around the prion field and wondering where can we have the greatest impact. And what we ended up feeling for our disease was that actually a lot of sort of basic biology building blocks had already been put in place. And a lot of different resources had sort of already been queued up. And there was an opportunity for us to sort of stand on top of those things and reach for a treatment. Try to use all that information to rationally develop a therapy. So we decided to do our PhDs in biomedical research and work in academic science to try to develop a drug. It wasn't immediately clear where and how to execute this next step. We were coming into the PhD process in this very unusual position as a husband-wife team with this really clear mission, and most entry-level scientists don't come in that way. And we weren't sure if we would find the right lab, if we would find the right mentors in the right place for us. It felt like a huge gamble. But we were super fortunate. We connected with mentors here at the Broad who saw that despite being as new as we were highly motivated to make this happen. And they actually supported us sort of from day one to come in and start up a new initiative and lead it ourselves, developing drugs for prion disease. So we have been here at the Broad ever since. We defended our PhDs. We rolled what we had built forward into what's now an independent lab that looks a little bit like this. We're now 6 members and looking to grow. And we've just had the tremendous privilege to be working on our mission all along. So we'll share with you today a little bit of what that progress has looked like. So starting from square one, what is the disease that we're trying to cure? When I was handed my test report, it actually didn't say prion disease. It said fatal familial insomnia, which turns out to be one of the historical names associated with prion disease. But there are many. These are names that were given in the clinic by different neurologists from different parts of the world who were looking at a patient and trying to name what they were seeing based on symptoms. We now know that underneath this sort of alphabet soup of all of these different names, there's a single gene, the prion protein gene, and there's a single protein. And that single protein is the cause of every single case. So I think we've been able to take this sort of seeming variability and compress it down in a useful way. Sometimes when people talk about precision medicine, you hear the objection, well, if we identify a different genetic driver of the cancer in every single patient, does that mean we have to develop a separate drug for each individual? Is that really realistic? I think this is actually an elegant example of how precision medicine can move us the other way. We take all these things that look different. We identify a single gene that unites all of them. And in doing so, we sort of consolidate our team around a shared goal. I think having this gene that unites all of these subtypes gives us a way to develop one drug for everyone affected. So what's a prion? When we talk about infectious disease, people think about viruses or bacteria. And while prion disease isn't an infectious disease, it is technically transmissible, meaning under specific circumstances, you can, say, transmit it from one animal to another. And this is one of the very first observations that was ever made about prion disease. So based on that observation, people thought, okay, surely this is a virus or it's a bacterium. And it is this invader from the outside that can come into the body and replicate itself and cause disease in that way. It actually took a long time and a lot of controversy for people to realize that that wasn't the case. And in fact, prion disease is caused by a protein that is not an invader from the outside. It's a protein that we all have in our bodies. And it's there in all of us right now, and normally not a problem. Normally, it's just part of being a mammal. But this protein has this unique liability. It's able to change its shape, sort of misfold into what's shown here in red, a disease-causing conformation. And the unique ability of that misfolded so-called prion is that it can grab onto other copies of the healthy prion protein and convert those. So it's sort of self-propagating in this way. It spreads itself through this domino effect across the brain and kills neurons, kills brain cells. So this was controversial in part because people wondered, how do we square this with the central dogma of biology? So central dogma, of course, tells us that we start from DNA, we start from genes, and from those instructions make RNA, which is the sort of messenger that carries those instructions to be made into a protein. The machinery of the cell, we have all these proteins that execute all of these different functions, and this is sort of a unidirectional process. But then how could you have a protein capable of making more copies of itself independent of DNA? So this was a big controversy and has been worked out, and we now appreciate that this doesn't circumvent the central dogma. It just adds this extra step. So prion protein is coming from DNA, but it's capable of flipping into this prion, which can grab more protein and convert that protein and cause disease. So that sort of genetic blueprint is really the orienting piece of information that we've built our mission around. We know the key steps in the production of this protein and in the mechanism of the disease, and therefore we can think about how do we intervene, how do we strategically intercept this process. And we have been very focused on the prion protein itself as a target. We know that you need the normal prion protein in order to have this misfolding and propagation of prions. You can think of that normal protein as the fuel to the fire, and without it, you can't have the fire. And this has actually been demonstrated in a series of animal studies that go back decades, showing that animals that don't have normal prion protein can't be infected with prions. They can't develop prion disease. So one of our goals is to develop a drug that reduces the amount of prion protein in the brain. And if you'd asked me on day one, I would have said, that is the goal. We just need to figure out what is the molecule that can do that. But one of the great surprises of being in science has been realizing that our mission is much bigger than we thought it was. I now have a much deeper appreciation for the complexity of drug development. And I think we actually need much more than just the molecule. There are all of these different areas that our lab has sort of grown into, where we have to build new tools, build new data sets, develop an understanding to assemble all these puzzle pieces into both a drug and a platform to meaningfully test that drug. So with that prelude, we'll take you through these areas sort of one by one and through the thought process and the troubleshooting and what this actually looks like day to day.
Thanks, Sonia. So I'll do this first vignette. So our goal in these is just to give you a sense of some of the data that we've collected and how we think through interpreting it and how that gets us closer to developing a drug. And so the first area is genetics. And our question here is, how can we identify who is at high risk for prion disease? Now, as background, analyses of death certificates have shown us that prion disease causes about 1 in every 5,000 deaths. So out of everyone walking out there on the street today, 0.02% of people will eventually die of prion disease. Now, bear in mind, not all cases of prion disease are genetic. The protein can also spontaneously misfold, even if you don't have a mutation in your DNA. The mutation just makes it much more likely that that'll happen in your lifetime. So from that baseline risk of 0.02% in a random person you pluck off the street, the question becomes, what is the risk in someone who has, for example, Sonia's mutation? And it turns out that there's actually two sides of this coin. So the other way you can ask that question is you can ask, how much more common is Sonia's mutation or any other mutation associated with prion disease? How much more common is it in cases, in people who die of prion disease, versus in controls in the general population? And it turns out if you can answer that question, then you can answer how high is lifetime risk for people with the mutation. The catch is you need to study a lot of people to answer this for a rare disease. And we just didn't have data on very many people until recently because DNA sequencing was expensive and only a few people had undergone it. But around the time that Sonia and I were joining the Broad in 2014, 2015, we were incredibly lucky. We got synced up with a group of Broad scientists who, at that time, had just completed compiling the world's largest database of human genetic variation. So we had access to a database of about 60,000 controls, people from the general population who had undergone DNA sequencing, and we knew the variations in their genome. And we were able to compare that to a database of a little over 10,000 prion disease cases, people who died of prion disease worldwide. And this is just a sampling of a few of the genetic variants that we looked at. So the way that we annotate these, you don't need to worry about the specifics, but basically each mutation is named after a position in the protein that's a few digits, and then the letters before and after tell you what amino acid changes into what other amino acid as a result of that mutation. But the point here was to basically zoom in on each population where these specific genetic mutations had been seen and say, how common is it in controls and how common is it in cases? And what you'll see here is that, look at this first variant, for example, E196A. It's actually about the same frequency in cases and controls, which suggests, as I was saying, the two sides of that coin. If it's no more common in cases than controls, that suggests that it does not increase risk of developing prion disease. People might have the same baseline risk of prion disease, 0.02%, as anyone on the street. but they don't have an increased risk. That mutation doesn't cause prion disease. And this was a huge revelation because until this time, people had, anytime you saw a mutation in a patient who had prion disease, neurologists and geneticists would just assume that that was what caused the disease. And so there were patients who'd actually been counseled, oh, wow, if you have this mutation, that's terrible news. You're doomed to the same fate as, basically the same news as Sonia got back in 2011. So when we went through all of these different mutations that people had observed in prion disease patients, we found that there was a whole range. So E196A is no more common in cases than controls. We don't think it gives you any increased risk of prion disease. Other mutations might give you a 1 or 10% lifetime risk of prion disease. And then there's this final category of mutations that, as far as we can tell, really are very common in cases and virtually absent in the general population. And so we think that they give a 90% or greater lifetime risk of prion disease. And I would have loved if the outcome of this study had been that we learned that Sonia's mutation wasn't as bad as we thought, but unfortunately, her mutation is D178N in that last row. So this just confirmed what we thought we already knew, which is that we do need to develop a drug. Sonia's mutation gives her a 90% or greater lifetime risk of developing prion disease. But for some other patients, this was really transformative. So there was a write-up in the Boston Globe about a month after this study came out about how a geneticist at Brigham and Women's, the same clinic where Sonia got her genetic test report, had recently seen someone who was at risk for the E196A mutation and had originally been told, well, if you have this, it's terrible news. And based on this study, was able to call that person back up and say, don't worry, actually, this mutation isn't a big deal after all. So that was really powerful to be able to change someone's life like that. And I think the good news, even for those of us who are still in the high-risk bucket, is that this allowed us to identify a patient population, right? By identifying who is at 90% or greater lifetime risk of developing prion disease, we've identified a group of people where the risk-benefit balance would be justified to enroll in a clinical trial, take an experimental drug. So that basically gives us a patient population in which we can start to think about how do we prevent the disease. So that was the genetics vignette, and I'll pass this back to Sonia for the next one.
Great. So long story short, we do need to develop a drug. And a big part of doing that is animal studies, which I'll touch on now. So I mentioned that our therapeutic strategy is to reduce the amount of prion protein in the brain. And as patient scientists, we are always asking the question, what is the most realistic way to do this in our lifetimes? One strategy that we're pursuing is to target the prion protein RNA. There are classes of molecules out there that can specifically target the RNA that encodes a specific target protein. The molecule that I'll talk about now is an antisense oligonucleotide, or ASO, that targets the mouse prion protein RNA and degrades it, resulting in less protein. And so we struck up a collaboration to test this kind of molecule in a prion disease animal model. We actually infect mice with prions. This is because prion disease is sort of a disease of mammalia. This is something we can do. We can actually model the disease, the whole disease process. And if you infect mice with prions, 5 months later, they'll develop full-blown prion disease. So this is quite a robust phenomenon. And in this study, we sort of queued up mice who would receive saline there in the top row, meaning salt water, no drug, or would receive that ASO drug, the drug that is lowering prion protein levels in the brain. And the variable was when would they start getting the drug. So some animals here, sort of moving down the table, got the drug before they even got injected with prions. Some got it right after that prion injection, or a couple weeks later, or several months later. And what we were studying here is what will sort of the differential impact be of treating at these different times? Because these times mean very different things in terms of what's going on in the brain. Have prions been introduced? Have they had a chance to cause brain damage? Have they had a chance to cause enough brain damage that you see symptoms? And when we look at the data that we got out of this study, you can see in that top row that the mice that didn't get a drug got sick right on schedule. So they developed 5 prion disease symptoms about 5 months after the prion injection. Then as we sort of move down the table, we can sort of group these 4 cohorts of animals together and say, wow, if the animals started getting treatment before brain damage was really detectable and really established, we had a big effect. We tripled the survival of these animals with this sort of early treatment. Then if we move further down, we get to the animals where brain damage was really well established by the time treatment began. And in these animals, we still see a benefit, but it's a much more modest benefit, somewhere on the order of 20%. So I think we learned a lot from this study. First of all, that PRP-lowering treatment really seems to work. And secondly, that when we treat matters a lot. So in those sort of late-treated animals, we do see much less benefit than those early-treated animals. And that has implications when we think about when can we catch patients and how much good can we do for them. I think this has been a big inspiration for us, thinking in the cases where we have the opportunity to identify people at risk before they're sick, we need to try to treat them before they get sick. So that creates a whole new set of mandates. And now I'll talk a little bit about natural history, which is sort of a fancy term for studying people who have a disease, or in this case, people who are at risk for developing a disease. So in Alzheimer's disease and other more slowly progressive neurodegenerative diseases, one of the big findings of the past decade has been that these diseases are sort of in process under the surface for decades, maybe, before you see symptoms. And if you have access to somebody's blood or somebody's spinal fluid, you can see these markers of damage that are sort of accumulating over time. But we didn't know if the same was true in prion disease. And so we launched a study to try to find out. We did this in collaboration with an awesome clinical research team at Mass General Hospital. And basically, the study is set up to bring mutation carriers, sort of in my position, healthy mutation carriers, and controls to Mass General Hospital annually. And people come, they fly from across the country. Some people have even flown in internationally. They spend 2 days in Boston. They do a whole bunch of tests. They get a lumbar puncture. They get a blood draw. People are frequently taking paid time off work to come participate in this study. So it's really just a tremendously generous thing that people are doing to participate and to help us build this data set. And while the study is ongoing, I'll share a snapshot of what we're learning from it. So here are 2 important markers that we look for in people's spinal fluid in this study. RT-QuIC is actually a diagnostic assay for prion disease. And it lets you really sensitively detect whether there are any misfolded prions in someone's spinal fluid. So if there are prions in the brain, some leak out into spinal fluid, and this is a way to detect them. Total tau is a marker of neurons that are dying. So if you have the sort of brain damage underway and you have neurons that are sort of bursting open, this is a protein that leaks out into the spinal fluid, and you can measure its levels going up. When we look at our non-mutation controls in this study, kind of as we expect, we don't see prions in their spinal fluid. They don't have prion disease. And when we look at their total tau level, it's in what has been reported as the normal range. So let's say 250. When we look at spinal fluid samples from symptomatic prion disease patients, who are sick at the moment of that lumbar puncture, we see a really high positive rate for RT-QuIC. So majority of them have prions detectable in their spinal fluid. And we see that level of that neuronal damage marker skyrockets. So it goes up more than tenfold. So the big question here was, when we look at our healthy controls, which of these 2 groups will they resemble? And this is the answer so far. So we've seen that the vast majority of these carriers don't have prions in their spinal fluid that we can measure. And we've also seen that their total tau levels are right there with the non-mutation controls. So what we conclude so far is that it looks like the disease process has not begun yet at the molecular level in the majority of these carriers. And I think that highlights really an amazing opportunity for us to catch people before that disease process has even begun and try to prevent it.
And that raises another question, which is, what do we then measure in those people, right? So you can imagine that if we had seen that people had dying neurons, maybe that would be a thing where you treat them with a drug and see if you can, reduce the amount of that marker. But if we're talking about people in whom the disease process hasn't even started yet, and we want to treat them with a drug and have some kind of evidence that the drug is working, that it's doing the job we designed it to do, we need a biomarker to tell us that. So a biomarker means a molecular measurement. It's not something that a patient can feel or that they could just tell you off the top of their head, is it high or low? But it's something that we can measure in a laboratory test that gives us information on what's going on in the patient's body. And in this case, the goal of our drug is to reduce the amount of prion protein in their brain, and the spinal fluid is bathing the brain. It's in direct contact. So is there a chance that we could measure the amount of prion protein in spinal fluid as a proxy for what's happened in the brain and use that to tell whether the drug is doing the job it's designed to do? And because everyone has normal prion protein in their brain, we should be able to do this even in healthy people where the disease process hasn't started and the misfolded protein isn't there. So we tried to get at this question through a couple of different animal studies. So one is using that same mouse model that Sonia described to you earlier. So these are mice that have a prion infection in the brain. And that means that they develop prion disease symptoms about 5 months later if you don't do anything about it. And we did a dose response. So we treated them with anywhere from 0 to 500 micrograms of this PRP-lowering drug. And the reason for giving that range of doses is it gives us a range of how much we lowered the amount of prion protein in their brain from down 6% to down 41%. And when we gave 2 doses of drug to mice that were infected with prions, again, the 0 dose, they developed symptoms about 5 months later, and we saw that the higher we dosed, the more we lowered PRP, the longer they lived. And if you translate that into a percentage increase in survival time in those animals, it actually lines up really well with how much we lowered the amount of prion protein in the brain. So even lowering it a little bit is better than nothing, but the more that you can lower this protein, the more good that you can do. So that gives us an idea that if we can measure how much we lowered prion protein, that will have implications for how much we will have benefited a patient. But there we're just measuring it in brain. The problem with mice is that their volume of spinal fluid is so small, it's actually really hard to measure prion protein in it. So to get at this question of does spinal fluid actually reflect what's going on in brain, we also did a study in rats. This is a larger animal, so the dose levels go higher, but it's the same idea. It's testing a range of doses to give us a range of how much did we lower prion protein in brain. Here we went anywhere from 0 to lowering it by 68%. And then we measured the change in prion protein concentration in spinal fluid in the same animals. And again, we see this beautiful correlation where the more that we lower prion protein in brain, the more it's lowered in spinal fluid. So together, all of this tells us that if we can lower prion protein in humans with an experimental drug, we should be able to read that out by doing a lumbar puncture, getting a little bit of that patient's spinal fluid, and measuring how much we've lowered prion protein in their spinal fluid. And so this could be a proxy to tell us, is the drug doing what we designed it to do? Is it working?
So finally, I'll talk a little bit about imagining the right clinical trials and talking to FDA about how to make those clinical trials happen. So if you picture what a clinical trial looks like, maybe you picture recruiting people who are sick, they have a disease, you give some of them drug, you give some of them placebo, and you watch to see who feels better or who functions better or who lives longer. And that is, in fact, the standard way that we do clinical trials. And I think we've queued up for you some of our hesitations about relying on that model exclusively for prion disease. One of the big challenges of this disease is that when you picture the sort of the tempo in the disease process, once the process starts, it moves so fast. So people are sort of healthy, 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 and then they're sort of falling off this cliff. And trying to catch them while they're sick is like trying to intercept this incredibly rapid downhill. I think to treat people on that downhill, what could we expect? If we caught them and managed to extend their life a few months longer on drug versus placebo, that would count technically as a positive result from the trial. If we caught them while they were sick and gave them a drug and managed to extend their survival in that state of impairment, that is a kind of result that can count as a positive result in a clinical trial. But is that the greatest good that we can imagine doing in this disease? What if we could leverage predictive genetic testing and the knowledge that we have about the at-risk community to treat people before they are on that downhill, before there's any sign of disease, and keep them healthy? When I think about the greatest good we can do, this is what I picture. And you know, it's not going to be possible in every case because, as Eric queued up, most cases of prion disease aren't genetic. And right now, we don't have the tools to identify people who are going to develop so-called sporadic prion disease until they have symptoms. For those people, I think what we need to do is push on diagnosis and try to catch them as early as we can. So that's one sort of frontier that we have to be pushing forward. But for people at genetic risk, I think our mandate is to find them before they ever get sick and treat them to try to keep them healthy. So I think it's sort of intuitive to all of us, even apart from the mouse data that we shared with you earlier, that prevention and treatment are not necessarily the same thing. And here's like a really common example of statins taken by millions and millions of people to reduce the risk of heart attack. And if you take them in a preventive fashion, they're very effective at doing that. But if you were to take a statin at the moment that you're having a heart attack, it wouldn't help you. So maybe prevention and treatment need to be addressed separately in this disease and maybe others as well. So as we've imagined what this kind of trial could look like, our goal is to reduce the amount of prion protein in the brain. We've seen some evidence that we can measure that reduction in spinal fluid. Could that reduction of prion protein in spinal fluid serve as the basis for getting a drug approved, maybe provisionally approved, to prevent this disease? The FDA actually has a pathway that's kind of designed for this, a pathway where your endpoint in a trial can be a biomarker that's quote unquote reasonably likely to predict clinical benefit, called accelerated approval. And the classical historical example here is measuring HIV viral load in order to provisionally approve drugs to treat AIDS. So in 2017, we went down to FDA for the first time to talk to them about whether the accelerated approval pathway could make sense in prion disease. We laid out our case for prevention. We laid out sort of our biomarker strategy and the fact that, you know, we have the single causal protein. Our goal is to reduce it. We can read that out in an accessible, you know, body compartment. Could that basic structure give us a path forward to test a drug for prevention? And there were 25 FDA scientists in the room that day. They were an amazing audience, actually. And basically what we heard from them is, this all makes sense. Of course you want to prevent this disease. Here you have a known biological pathway, and you know where you're trying to intervene, and you know how you can measure that. So this makes sense. But there will be a lot of homework, and we will help you figure out all the right experiments to do, but there's going to be a lot of data to gather in order to make this unconventional path possible. So that is a lot of what we continue to do in the lab as we hopefully move towards clinical trials.
Of course, nothing happens without patients. And one of the challenges is, you know, if you're thinking about sick patients, they're seeing a neurologist. Their neurologist sends a spinal fluid sample for testing to a prion center. There's a way to find those patients. Whereas for asymptomatic, healthy, at-risk people like Sonia, they're probably not seeing a neurologist regularly. So one of the things that we did in collaboration with Broad's data donation platform is launch a registry. So since 2017, with this launched, we've enrolled over 200 people who self-identify as healthy but at risk for prion disease and are interested in participating in research. So we think that we have identified a patient population that's ready for clinical trials, and there's really a lot of demand, and we hear all the time from people that they want an experimental drug. They want any shot on goal that they can have at preventing this disease. So what does the path ahead look like? So the program that Sonia told you about, the experimental drugs that we tested in mice and rats, we partnered with Ionis Pharmaceuticals, a company in California. They're developing basically the human version of that molecule, and the latest word that they've told the patient community is that they think they may be able to do the first in-human dosing study later this year, in 2022. And that their goal is to develop this drug both for prevention in pre-symptomatic patients and for treatment in symptomatic patients, and have those as separate pathways, which we think would be great. So that seems like a long ways to have come. I mean, it's really exciting for us after all of this work to have the prospect of actually having a drug in human trials. But it's important to remember that in many ways, that's just the beginning, right? A clinical trial itself is just an experiment to see if the drug works, and there's no guarantee that it will. But whether or not this particular drug succeeds or fails, I think that a lot of what we've built here is forever, right? Assembling these puzzle pieces, identifying a patient population, a therapeutic hypothesis, right? This idea that if we lower prion protein, that will delay the disease, the biomarker, the idea that we can measure whether we've lowered the protein. All of those pieces will continue to matter. And if this drug doesn't work, another one down the road will. So we have a lot to be optimistic about, but the question for us is always, how do we get there even sooner? Thank you so much for listening to our story. If you feel inspired to support us, you can go to prionalliance.org/donate, and we're really happy to take questions. Thank you so much.
Thank you, Sonia and Eric. What a great talk. And we are getting questions from the audience. I'm seeing a lot of questions about therapeutics, so we're going to start at that point. You all talked about ASOs as a therapeutics pathway here. Are ASOs being used for therapeutics research for other disease areas, maybe other rare diseases? And if you could share a little bit more about that.
Sure, absolutely. I mean, the short answer is yes. So ASOs are being explored for a range of different diseases. A sort of success story that we point to often is that there's an approved ASO therapy for spinal muscular atrophy, which is a rare neuromuscular disease that, in its most extreme form, 10 years ago, if a child had this disease, they might not make it to the age of 2. So really severe disease. And their ASO works a little bit differently from ours. It seeks to increase the expression of a particular protein rather than decrease, like we're trying to do. But it's been a really successful therapeutic. It has actually inspired sort of a wave of therapeutics. There's now more than one approved drug for spinal muscular atrophy, which is incredible. So we really look to them for inspiration. But there's a range of other ASOs under development for brain diseases, also for non-brain diseases, for rare and for common. Any examples you want to highlight? No?
Just kind of going back to the process of identifying ASOs, how did you find this particular drug candidate? And at that time, before you finalized on ASO, did you have other candidates? And anything that you could comment about the types of mechanisms?
Well, we can't take any credit for discovering the actual molecule, so that was done at Ionis Pharmaceuticals. They developed both the sort of tool compound that we use in mice and rats, and they're developing the drug that'll eventually go forward to humans. This came about as a result of us approaching them in 2014. So at that time, nusinersen, their drug for spinal muscular atrophy, was in clinical trials. They had just figured out that they had a way to dose these compounds into spinal fluid, where they thought they were actually getting good drug uptake across the brain. And so even though ASOs had been around for decades before that, it was never clear that you could actually use them in a brain disease until right around that time in 2014. So I think before 2014, we had looked at, there's a lot of work on small molecule drugs, so basically think pills in your medicine cabinet. there had been academic groups that had been experimenting with small molecule drugs for prion disease, with some success in mice, but a lot of really significant barriers that looked like it wasn't going to be able to make the leap to humans. And we didn't really know where to go from there. And the fact that ASOs now seem to be a workable class of drug for the brain was really exciting. And so we went out and basically pitched our disease to Ionis Pharmaceuticals and showed them some of the same introductory stuff that we showed you today. But obviously, we didn't have nearly as much data back then. And they really worked with us. They said, here's the package of data we would need to assemble together with you in order to make this a viable option. But fast forwarding to 2022, I don't think it's by any means the only viable option. I think a lot of new drug modalities have come along since then that have the hope of potentially being able to address this as well.
Since there's just generally a lot of interest in this area, I'm going to ask a couple more questions about the drug discovery path. How far can the protein be lowered without impacting health, both short and long term?
Yep, it's a really good question. And as far as we can tell, we're actually quite lucky in the drug target that we're up against. We have a lot of evidence that it's essential for disease, and we also have a fair amount of evidence that it's expendable for health. So that comes from different systems. We have a lot of different knockout animal models, so animals that have been engineered so that they don't express any prion protein, and people have been studying those animals for a long time. Basically, there's sort of one change in these animals that has been sort of replicated, and it's minor and age dependent. It looks like they maybe have less sensitivity in their peripheral nerves as they age. Even having one copy of the prion protein gene actually rescues that minor sort of symptom. And so we think, it's only been seen in animals that have like 0 prion protein. Right now, we don't know of any drug that could get you to 0. I think in a human, what we're aiming for, what would be amazing, is if we could reduce prion protein by 50%, and maybe in the future we can aspire to do more. But we're not talking about a total knockout situation. So I think the animals, by and large, give us no reason for fear. There's also knockout cows, there's knockout goats, they're healthy. And then we also have, and this is one of the first studies that we were able to do, we used that big human genetic data set that Eric was talking about. And one of the things we saw in this very large number of sort of humans that we were able to look at their exomes or part of their genomes, what we saw was some people who happened to have an inactivated copy of the prion protein gene just by chance. And you can look at sort of across the genome, and Eric has led a big study looking at a range of different genes. You can ask the question, do I see inactivated copies as often as I would expect to based on chance? If you don't, then that might suggest that natural selection is acting against that sort of inactivation. But in the case of prion protein gene, we see just as many inactivated copies as we expect. So that provides some human genetic evidence that you can have reduced dosage of this protein and still be fine. And we actually have information on some of those people sort of giving us a snapshot of their health, and they're healthy.
Thank you. This preventative approach that you are developing with PRP lowering treatments for prion, do you all hope that a similar treatment could be developed for other neurodegenerative disease such as Huntington's? And what are some possibilities that you imagine with the same mechanistic pathway?
Sure. I mean, I think we and others are really interested in this possibility, and sort of broadening out from ASOs and looking at the different classes of therapeutics that can target disease-causing, sort of an RNA that encodes a disease-causing protein. I think there are a lot of different biotechs and companies out there that are looking at Huntington's and lowering the amount of Huntington protein by targeting the RNA. There are a lot of companies looking at ALS and lowering proteins like SOD1 that are causal for ALS. And I think what unites these diseases is that they're all, quote unquote, gain of function, and they're all there are monogenic forms. So gain of function meaning you have a protein that's going wrong, that's the cause of disease, as opposed to not having enough of a protein, and you want to lower the amount of that problem protein, and identified single genes that you could really target. So I think where those sort of factors align, this strategy or like allied strategy is absolutely going to be explored.
That's great. I'm going to switch gears a little bit, and I want to applaud on the amazing scientific career journey that you shared with the audience today. And to that light, I think my question was around when you were setting out on this journey, like you were changing careers from completely different fields to starting to train as a scientist, are there certain challenges you experienced, or like were there the systems or people who told you that this process will be challenging? If you can share a little bit about that experience, because you kind of came into a PhD program from like being professionals in other fields and then started out on your own discovery path. So there are a lot of people who are attending today who are students, and I think this journey, sharing about this journey will be very helpful for them.
Sure. You want to take a stab?
I don't know that there were any human enemies. The villain in this story is really prion protein. I do think that there were people who cautioned us. One of the pieces of caution we got was, do you really want to think about this all day? Will that be too depressing? And I think maybe if we were neurologists seeing patients in the clinic, it really would be too depressing. But I think thinking about the molecular side of the disease in a biomedical research setting is very different. And I find it really energizing. I don't find it depressing at all. I think another word of caution we got was, don't you know that making a drug takes 15 years and $10 billion, or various really terrifying figures. And indeed, here we are 10 years on and don't quite have a drug that's been tested in humans yet. So it is a really long journey. But I think we were blessed with the gift of time, right? Having a predictive genetic test report and having a lot of advance notice on the problem you need to solve gives you options you might not have otherwise. In general, I think people have been just enormously supportive, especially here at the Broad Institute. I mean, every conversation, every scientific talk that we go into, we lead with our personal story, and people really respond to that. People really want to help. They're really moved and energized by that, and I'm just so grateful for all the help we've gotten from basically from every angle, from every source.
Thank you. I do have a question about someone in the audience is asking, is the asymptomatic person that showed prion levels in your trial someone that you catched actually just starting to develop the disease early on, and are you able to follow this particular patient along?
Yeah, I mean, I think that's a reasonable hypothesis, and this is something that we're still following up on to sort of understand the prognostic value of that information. Our study is designed to sort of follow people year by year and have interim check-ins, and so we hope to see, what we hope to see is our participants living long and healthy lives, but we hope to be able to follow everybody and learn from what we see. And we hope to be able to enroll these people in a trial for a drug that is effective, as soon as we can.
Sonia, I'd like to go back to some of the basic biology slides that you were sharing, and you shared about the prion protein. What's the function of the protein when nothing goes wrong? Like when the misfolding is not happening, what's the general function?
Yeah. It's a good question. It sort of goes back to what I was saying earlier about knockout animal models. That's one of our best tools for sort of understanding native function. What's the normal job of a protein? And so people have looked really hard in, for example, mice, to understand if they don't have prion protein, what's missing, what's different. I will say the big thing that's different is that you can't infect them with prions. And that was like the first and second and third thing that was written up about these mice. But people kept looking and kept thinking, there must be something else. So one thing that's emerged is that possibly a fragment of the normal prion protein gets cleaved off and goes and binds a receptor that promotes myelin maintenance on peripheral nerves. And myelin is sort of a sheath that's wrapped around the nerve that helps it conduct a signal fast enough. And in the absence of prion protein, in these knockout mice with zero prion protein, that sort of age-dependent subtle change that I mentioned earlier, the fact that they seem to have a little bit less sensitivity maybe in their pause, could be due to that event not happening, the myelin sort of not being supported in this way and having slightly different properties. So that could be one normal function of prion protein. I don't know if it tells us the whole story. We know that there's a lot of prion protein in the brain, and I think that might not tell us why. We still have yet to find phenotypes, like something that prion protein is responsible for in the central nervous system directly. But I think we don't at all assume that just because we have a protein, it's like essential to have it your whole life long at those exact levels, I guess. And biology, like all the lines of evidence we've been able to bring together so far suggest that whatever prion protein's job is, whatever the whole story is there, maybe there are other proteins that also support those functions. So when you subtract prion protein, you don't see big changes. Maybe it's just not an important job. So people will be monitored really, really closely for safety in these clinical trials. And the drug types that we've talked about today are all reversible. So you dose and then you wash out. And if there were to be any side effects, you would wash out the drug and start again at a lower dose or with a different type of drug.
Thank you, Sonia. Thank you, Eric. Thank you both for a wonderful talk. And thank you to all our viewers today for joining us virtually this afternoon. We hope you enjoyed today's talk. If you liked what you heard, then consider watching other talks from the Science for All Seasons series. You can find them all at broadinstitute.org/SFAS. And consider joining us again on August 2nd when Heidi Rehm, a Broad geneticist, will deliver this year's Ileana Hector Memorial Lecture and talk about the clinical use of genomics. A recording of today's talk will be posted to our YouTube channel at youtube.com/user/Broad-Institute. To find out about other live stream events and new videos from the Broad, consider subscribing to our YouTube channel and tell your family and friends to do so. Thank you so much for joining us this afternoon, and have a wonderful day, everyone. And thank you, Sonia and Eric.
Thank you.
Thank you.